According to a recent study, the way your muscles communicate with other tissues in your body is determined by your genetic sex. Greetings, everyone. Today's video centers around a fascinating study featured in eLife, which sheds light on the impact of genetic sex and sex hormones on muscle signaling to other bodily tissues. Myokines, which are proteins secreted by muscles, play a vital role in numerous physiological processes. The study discovered that these signaling pathways for myokines differ between sexes and are influenced by hormones, particularly estradiol. This understanding holds great importance for the study of metabolism and metabolic disorders. Moving forward, researchers will delve deeper into investigating the effects of hormones. So without further delay, let us dive into our discussion. In a groundbreaking study published in eLife titled "Genetic Variation of Putative Myokine Signaling is Dominated by Biologic Sex and Sex Hormones," researchers have for the first time examined the impact of genetic architecture on muscle signaling to other tissues, emphasizing the crucial role of sex and estrogens in these processes. According to Marcus M. Seldin, PhD, an assistant professor of biological chemistry at UCI School of Medicine and senior author, muscle plays a crucial role in maintaining metabolic state, and any impairment in muscle function is a defining characteristic of diseases like obesity, type 2 diabetes, and cardiovascular disease. Myokines, proteins secreted by muscles, have the ability to interact with various tissues and play important roles in numerous processes. This communication between skeletal muscles and organs like the kidneys, liver, and brain is crucial for maintaining metabolic equilibrium within the body. Myokines are involved in a range of functions, including inflammation, cancer, exercise-induced changes, and even cognition. Despite the undeniable significance of myokines in various physiological outcomes, there is still a lack of understanding regarding their regulation and effects. In this particular investigation, the team of researchers conducted a comprehensive examination of genetic correlations pertaining to the regulation of myokine genes, the composition of muscle cells, cross-tissue signaling, and the interaction between genetic sex and these factors in human subjects. While the levels of myokine expression and the proportions of different cell types within skeletal muscle displayed minimal disparities between males and females, the majority of significant cross-tissue connections operated in a manner that was specific to sex or dependent on hormonal influences, particularly estradiol. These sex and hormone-specific effects were consistently observed across various crucial metabolic tissues, such as the liver, pancreas, hypothalamus, intestine, heart, and visceral and subcutaneous adipose tissue. Notably, this study shed light on specific instances where muscle communication with the pancreas was more pronounced in females, whereas the liver played a dominant role in males. According to Seldin, the significance of skeletal muscle in maintaining physiological balance is already well known. The objective of this research was to explore the interactions between muscle and metabolic tissues, while emphasizing the need to take into account genetic sex and sexual hormones in metabolic studies. In the future, the research team intends to create cell-based systems to assess the newly discovered hormones from this study and explore the reasons behind their differing signaling mechanisms in males and females. The National Institutes of Health provided the necessary support for this project. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.